Hello, this is Scott Carpenter, and this is part 2 of the DNA study in a nutshell. It's been a little bit. I had food poisoning last week. It was a rough week. Ugh, I'll tell you what, I'm almost 60 years old, and I had food poisoning back when I was in the military in the Air Force, and that would have been like in 1983, 84. Been in early 84. And I remember being sick as a dog and having to crawl everywhere and being real nauseous. And the second bout of food poisoning didn't disappoint. I'm telling you, that was I had just finished a video and was rendering it, and I was sitting here at my desk. I stood up and the room started spinning. I thought I was having a stroke. Went and ran to the kitchen and took me a couple of aspirin, and then I started feeling the cramping and the grumbling and the nausea, and I made it to the bathroom, and 8 hours later, I was in bed, passed out. So it's like it punched me in the gut pretty hard. I gotta admit that. I'm not over it yet. I mean, I went back to work on Friday, and I'm doing things, but I'm definitely way far from 100%, and doing what I should be doing with the fluids, and still can't eat anything more than chicken and grilled chicken or chicken and chicken noodle soups and crackers, and I tried to do some eggs yesterday, just a couple scrambled eggs, and that was really, that gave me some really bad indigestion. So not quite there yet on completely back, but we're getting there. So there's an update on me and why it's been just a little bit since I posted. And so in part 2, let's see. Let's go back. Yeah, let's go back. Processing the hair samples. I think I left off with, this is some of the different samples they had. And sample 26, and this may come into play when the trolls and the detractors begin attacking, so it's good to know this information. But sample 26 was flesh and hair. And so they sent a piece of the flesh off to Douglas Toller, MD, and he is a pathologist, and he determined the skin was not human, and that the tissue was not contaminated with bacteria or fungus, which was important. And so the skin was not, rotted, it had bacteria or fungus, and it was not human. Several hairs with the follicles from sample 26 were sent to the Southwestern Institute of Forensic Sciences for analysis, and the hairs were processed robotically, and so that means no human contamination and no touching. And then a core sample from the flesh was also taken to ensure no contamination, and this sample contained a high quality DNA that yielded both mitochondrial DNA and, more importantly, nuclear DNA. Because we have, had a bunch of mitochondrial, but now we're getting down to where we're actually getting nucleosides, the nuclear DNA, which comes from the father. And the nuclear DNA was of high quantity and quality. The core samples underwent a structural analysis using an electron microscope. So let's go to the next slide. Sample 31 was blood from a food trap. This was from the Erickson study up in Kentucky, and they did a real interesting thing. Is they took sandpaper. They had a Sasquatch, the Matilda Sasquatch. It was a juvenile female, and she really liked pancakes and syrup, and so they took a paper plate, and on the bottom of the paper plate they glued sandpaper, gritty sandpaper, and then they put the pancakes in the syrup, and when she got, she lit the plate, and when she did, the gritty sandpaper, cut her tongue a little bit, and they were able to get some blood from that. And that's sample 31 was collected from a research site using food trap and forensic techniques to ensure there were no human contamination. The DNA extracted from the blood was high enough quality and quantity for a complete nuclear genome. So they got a full nuclear genome from sample 31. So you had the hair sample. The previous hair sample came from California, the flesh and hair, and this came from Kentucky. And the person, they had actually hired a PhD scientist to assist with the collection of the DNA and set up the food traps, and so it was forensically collected by this person. And the sample 140 was the fresh dried blood on the downspout, and you saw that picture last time. For some reason Sasquatch got mad, grabbed the downspout and literally bit it or chewed on it. And the DNA extracted from the blood was high enough quality and quantity to get another complete nuclear DNA. So they got 3 different samples, 3 different locations across the United States, and they were able to get nuclear DNA complete. So this, now I've done this before, but I think it's important because when a lot of people just gloss over, their eyes get all, oh God, there's detail here. I don't want detail. I'm scrolling my feet. But when the detractors began to attack the study, back when the study was going on, and now what their tactic is going to be is they're going to rely on your ignorance and the general population's ignorance of DNA, how DNA samples are processed, and how it all works. And they can use that ignorance of that process in DNA to pull the wool over your eyes, fool you, lie to you, misrepresent the truth, and they'll do it all. And I've seen it multiple times. And they'll mischaracterize something, or they'll use your lack of knowledge of this DNA process to say things that, since people don't know what they're talking, how DNA works, they'll just say, oh, I guess they're right. So real quick, and for some this is old hat, for others this is, I think this will be a good little boot camp. So DNA has a double helix shape. It's like a twisted ladder or spiral. There are 4 nucleotides in the ladder: adenine, thymine, guanine, and cytosine. And the nitrogen bases are found in pairs. A only pairs with T, and C only pairs with G. The bases are held together by hydrogen bonds. If you unzip the DNA so you can have one side of the ladder, you still know what the corresponding nucleotide is, because DNA can only pair in this order. This is important to understand for discussions later in this chapter. In other words, they can only pair A with T, C with G. So if you have one side, you know what the other side is automatically, because of if it's an A and a C, then you know the other side is a TGG. I mean, that's just, you know, because they pair. And so here is a graphic, and you can see here, this is a graphical representation, and the sugars and phosphates that hold everything together, and you got a hydrogen bond between the nucleotides. I'm still not 100%, so we probably won't go long on this video. There are two types of this is important. There are two types of DNA, nuclear and mitochondrial. The mitochondrial DNA is found in the cell mitochondria and is from the mother only. The nuclear DNA is found in the cell's nucleus and is a combination of the genetic code from both the mother and the father. Mitochondrial DNA is more abundant and easier to extract and test, while nuclear DNA is more difficult to extract and test. Samples in quality and quantity are required for successful extraction of nuclear DNA. And of course, we just went over definitions and terms. Mitochondrial DNA is found in the cell mitochondria. It's from the mother only. Nuclear DNA, father and mother in the nucleus. GenBank, this is important. You may hear this thrown around, but what are they talking about GenBank? GenBank is an institution that collects and stores DNA sequences for all life forms on the planet. Once a DNA sequence is placed in this database, comparative analysis can be run on it using a special software called BLAST. The BLAST software is a software engine that compares a DNA sequence against all the DNA sequences stored in GenBank database and reports the matches from this database. So basically, it's a search engine, and if I've got a subscription, I'll have my BLAST browser or engine on my laptop, and I'll have my sequence. I'll have run some DNA, and I'll get a sequence. It's a digital file, you know, and I'll have my sequence, and I can take that sequence, load it, and then I can log into GenBank and have BLAST do the comparison against that database. Now, it may take a long time, and some of these samples, depending on how large, how long your sequence is, the base pairs, it could take quite a while. And the time I wrote the book, the current number of base pairs stored in GenBank is 152,599,230,112. I did that right. So here's a representation of the mitochondrial versus the nuclear. You can see you have a man and a woman. The woman, it says out here, is the mitochondrion, and so that's where the mitochondrial DNA is. Here's the nucleus, and a combination of the two are in the nuclear, and that's the nuclear DNA. For example, you know, I was saying here, it is possible to have mitochondrial DNA from one species and nuclear DNA containing genetic material from an entirely different species. For example, a mule has a donkey father and a horse mother. Therefore, it has horse mitochondrial DNA, and will have a combination of donkey and horse in the nuclear. Since I just butchered that. So basically, a mule, which is a cross between a donkey and a horse, the mitochondrial DNA, if the mother is a horse, then the mitochondrial DNA will be horse. And there'll be a combination of donkey and horse in the nuclear DNA. That's a mouthful. So how do they perform a DNA test? And so, you know, you've got to in order to actually sequence out the DNA, it's tightly wound together in its natural state. It's just it's like a jumbled wad of rubber bands. And in order to get the sequences out, then you got to unwind these sequences, and you got to unzip them. And remember, you only need one side of the helix or the ladder to decode the DNA. as I've previously pointed out. It's A with T and C with G, and they're always paired. So if we have one side of the ladder, we know what the other side has to be. So basically what they do is they like to take a blood sample, they'll extract the DNA, they'll enzymes cut the DNA, and then the DNA fragments are separated by size in the gel, and the bands are stained to make them more visible. And they can do this by machine now. They have machines that do this, and special enzymes are used to break down the DNA and unzip them into single strands. Once we have the DNA broken down into strands, we need to stack them like strands together. There are multiple copies of the DNA in the cell. So you've got all these multiple copies of the same DNA floating around. So you unzip them, break them down into smaller strands, and then you need to stack them one on top of another so they'll amplify, so you can actually see them. So it's just it's kind of like a transparency. If it's like you're laying one transparency on top of the other. If you only have one copy of the transparency, God, it is light and difficult to see. But if you have 25 copies of the same transparency, then you can stack them together one on top of the other and you can see the picture clearly. DNA testing is the same way. The greater the DNA density, the better this will make the DNA easier to see, and this being able to see is what is called amplifying. And of course, in order to stack the DNA, the scientist will use what is known as a primer. The primer's basic function is to gather like strands of DNA together and stack them so it is easier to see the DNA sequence or amplify the sequence. In the early days of DNA testing, primers had to be designed for individual species and even subspecies with a family of organisms. For example, primers were developed for humans, bears, dogs, cats, pigs, cattle, etc. If you were trying to identify an unknown DNA, you would use a primer from the individual species on the sample. If the primer found and stacked the DNA together or amplified the DNA, then you knew you had the species. In other words, back in the day, you had an unknown DNA sample. You'd have to run a primer for dogs and see if it amplified. If it didn't amplify, then you'd try cats. If it didn't amplify, you'd try pig. If it didn't amplify, you'd try bear. So forth and so on. When it did amplify, then you knew, okay, well, I had my primer for a bear. It amplified, it's a bear. These primers still exist today, but as scientific advancements have been made in the field of DNA processing machines, I'm running on that sentence. The primers still exist today, but as scientific advancements have been made in the field of DNA, processing machines were developed that could process the DNA faster and more accurately. Primers were also developed to help speed up the process. What is known as a universal mammal primer was developed. Scientists, with the use of a DNA sequencer and universal primers, can now process DNA and get a profile faster and more accurately. Just like with the species-specific primers, if you use a mammal primer and the DNA does not amplify, then your DNA sample is not from a mammal. Here is a very important aspect to the DNA testing, and this does come into, and there were several times when the detractors of the study used this information here to confuse people, and when they're talking about the DNA study, and sometimes it's what's not there that's just as important or not more important than what's there. And so they kind of played fast and loose with the wording here in the study, when they were doing their critique of the study. So this is important. Just like the species-specific primers, if you use a mammal primer and the DNA does not amplify, then your sample is not from a mammal. Here is a very important aspect of DNA testing. The test will be affected by the quantity, quality, and purity of the DNA sample. If you have multiple copies of the DNA strands, the strands are not damaged and intact, and the DNA is all from the same organism, then the primer will amplify correctly and you will get a clear result. If you have a limited amounts that are contaminated, the results will not amplify very well. To use the transparency analogy again, now if you have two transparencies of two different pictures, some parts of the picture may be alike and some different. So the primers stack them on top of each other and the picture now is fuzzy or blurred and not easy to recognize. And that's how you know if you've got a contaminated sample, or a sample from, and that means from multiple organisms. In other words, two different dogs, two different cats, or whatever. The process I've just described is a very simple language. It's known as the polymerase chain reaction, PCR test. And in this day and age, everybody's heard a whole lot about PCR testing. This is the method the independent reference labs and Dr. Ketchum used in the DNA study to sequence the Sasquatch DNA. They used the PCR testing method in order to amplify and sequence the DNA. So here, I may want to skip over this. This may be getting too much into the weeds here. This is just talking about non-human. What is important here is that non-human hair yields human mitochondrial DNA. I said it's not important. It is very, very important. In fact, that's one of the mind blowers of the study. And if you take away anything I've said today, this is what you need to take away. When they were testing non-human hair samples, in other words, they were Sasquatch samples, they yielded human DNA. In other words, so they looked at the hair and they're like, this is not human hair, but it has human DNA. So it's talking about, let me, yeah, just basically, you can read it if you want to. It's just talking about, this is the detail on the different, what a human hair looks like, and what they call it novel, which is unknown or new, and that's what novel means. And it's the novel hair, or the novel structures, is Sasquatch. The hairs were evaluated for DNA testing by observing the presence or absence of hair roots and adherent to tissue materials. Hairs with apparent translucent tissue material and or an anagen or catagen phase roots were considered for suitable candidates for nuclear DNA testing. Hairs with telogen phase roots without tissue or hairs lacking roots or tissue were considered suitable for mitochondrial DNA testing. Hairs that exhibited non-human microscopic characteristics and that did not match any known animal species were recommended for testing, for DNA testing. In other words, non-human hairs and hairs that did not match any known animal. So this is an actual picture of a Sasquatch hair under a microscope. That's what it looks like. And that was from the DNA study. So the smoking gun, David Pilates, my good friend, he did an article and talked about how for years, from the 80s, 90s to or 2000s, people would see a Sasquatch. You'd go over, let's say, go over a fence row, and get some hair caught, and on the fence row they'd go over and they'd get the hair and they'd send it off and have it tested. And in the test, the mitochondrial DNA would come back human, and then the lab would declare, well, it must have been contaminated. In other words, and we've had this, years of this, like hundreds of samples with witnesses, with the, chain of custody. As far as when I mean chain of custody, I mean person sees it, it leaves the hair, there were witnesses, at least one, sometimes multiple witnesses, and it was definitely Sasquatch hair, and then it comes back human. And instead of saying, hey, we've got a human hybrid, or we've got some interesting results where this hair, returned, human DNA, maybe it's a hybrid, they would always throw up their hand and say, oh, It must have been contaminated because we know it can't be human. So yeah, it can be, and it is. But you know, this was just years and years of this. I mean, you know, yeah, and the other thing would always come back would be the old unknown. It was an unknown mammal. Sometimes they would throw that out there. So you can see here human hair compared to at the top here and Sasquatch here, and you can see the difference in the hair. The hair samples were tested using universal mitochondrial DNA cytochrome B primers for species determination. So they're trying to figure out what species it was. All 111 hair samples screened revealed 100% human. One more time. All 111 Sasquatch hair samples revealed 100% human cytochrome B. Furthermore, no heteroplasmic bases were found that would indicate contamination or mixture. That fancy word, heteroplasmy, is defined as the presence of more than one mitochondrial genome. That's where, you know, I told you about mixing. You know, that would be contamination. In words, if I touched it and they didn't clean it well, then my genome would be mixed in with them, so it'd be mixed up. So that wasn't present. The presence of more than one mitochondrial genome with a tissue sample from a single individual. No heteroplasmic bases were found in the mitochondrial DNA was from a single source. Therefore, contamination is impossible. I'm going to hammer that because that's exactly what you're going to hear over and over again. It's contaminated. It's contaminated. It's contaminated. Sorry. Another interesting thing, how much time? I'm going to 26. Let me see if I can get through haplogroups because this is interesting. Because the haplogroup tells you where the origin of the origin of the DNA came from, on the Earth, and it's used to. Oops, I got my. Sorry about that. To put my hand too close to the camera. There, I'm coming back into focus. So basically, the haplogroup is the group of genes that a child inherits from the mother. Like haplotypes are grouped together and assigned a letter from the alphabet. This group is called a haplogroup. HV region sequencing will give the haplogroup of the individual. So and that tells you the region of the world it came from. You know, they use this in this DNA testing online that, you know, so you can figure out where you came from. So 30 hair samples that had ample skin tags were selected for mitochondrial DNA complete genome sequencing and for the HV region sequencing to determine the haplogroups of the samples. These samples were sent to Family Tree DNA for sequencing. Family Tree DNA does, DNA, haplogrouping, for people to trace their lineage. The source of the samples was held, so it was a blind study. They didn't know what they were testing. They were just given the studies and said, hey, test these. Give us the haplogroups. The DNA samples were successfully amplified, this is from the study, and sequenced across the whole mitochondrial genome and the HV1 locus using both human-specific and universal primers. The sequence were subject to blast searches in the GenBank and show consistent homology with human haplogroups. No mitochondrial DNA homology with apes, Neanderthals, Denosia, cave sequences were found. In other words, they ran these blasts, they blasted this DNA, in other words, they compared it to the GenBank, and it did not match apes, it didn't match Neanderthal, it didn't, or Dysnovia, cave sequences were found, Dysnovians. So it wasn't any of those species, because a lot of people trying to throw that in there. The results showed that the mitochondrial DNA was human. The haplogroups were human. Blast searches showed the sequences did not match. And I just said that. The 30 of the 30 samples, 20 yielded whole mitochondrial sequences and 10 yielded partial mitochondrial genomes. The 30 samples showed 16 different human haplotypes, so 16 different haplotypes. The interesting and unexpected finding was the origin of the haplogroups. The majority of the haplogroups show origination from Europe, Asia, and Africa. Only 4 of the haplogroups originated close to where the samples were collected in North America. See, everybody was thinking, well, you know, these Sasquatch, they'll have Native American human. That was what the cross was. That was what the assumption was from a lot of people. But in reality, that's not the truth. That's not what happened. All right, I'm getting ready to have to break this. I'm up to 30 minutes, so I'm going to reset. Okay, I'm back. This camera breaks it at 30, since it's high def, it's 4K, it'll break it at 30 minutes. And I need to, I'll wrap part 2 up. But this is really, really important and very interesting. Again, the origins of these 30 Sasquatch were from Europe, Asia, and Africa. And a lot of them, when it says Asia and Europe, a lot of them came from the Middle East. A bunch came from the Middle East. Here's the map here of those samples. And you can see here, the red dots are where they collected, and the squares are where the haplogroups are around the world. Here you got the three different groups out of North Africa, and a couple in Israel in that area, across Russia, a whole bunch in Europe. I said a whole bunch in Israel. I mean, in around the Holy Land. I meant to say a whole bunch in Europe. Sorry. It's kind of obvious when you look at the map, but I stand corrected. But you can see it. You know, here's where they were found, and here's where they came from. And I think that's a good place to stop with the haplogroups. And I know this is sometimes mind-numbing, but I think it's important if you're interested in the study and what's going to be coming up soon. It's a good primer to have, and also it arms you so you'll be knowledgeable of what's going on. So when you see misinformation, disinformation from different individuals and groups, and they try to mislead you and confuse you, you'll know, you'll be better equipped to ignore that and understand what they're trying to do, and not let that get you sidetracked. And so, my apologies if I've not been just spot on. Like I said, I'm just still working on getting over the food poisoning. Lost over 10 pounds in a week, and so it's pretty rough. And so anyway, enough about my troubles. Hope you enjoyed it. God bless everybody. Y'all have a safe week, and we'll be back with part 2 here. Part 2. We just did part 2. We'll be back with part 3 here pretty soon. Y'all take care.